Hi. We're continuing with my notes for life science and cell biology. Um, in a nutshell, um, we have reached the section of learning about DNA and the molecule and the structure of it, which is the genetic material inside the nuclei of cells. Um, Structural proteins, as shown here in the 3D model, um, organize the DNA into a compact structure called chromatin. These proteins organize the DNA into a compact structure. Um, okay, let's see. These are called histones, these proteins. And in prokaryotes, there's numerous types of proteins involved for the same purpose, organizing DNA material. So DNA interacts. DNA in the diagram is orange, the helix structure, and the histone proteins are in blue, surrounding. So the protein's basic amino acids bind to the acetic phosphate groups of DNA. So the basic aspect of the amino acids of the proteins attracts to the phosphate groups of DNA molecules which are acidic. And that's how they're able to interact. Um, an example, the lambda repressor helix turn helix transcription factor binds to its DNA target. So this is one type of protein. The restriction enzyme EcoRV in green, the protein gets into a complex with its substrate DNA. So that's another protein that binds with DNA. So these different kinds of proteins control the transcription and the expression of the DNA material molecule. They control various aspects of how it is turned into RNA and how it is expressed, etc. Enzymes called DNA ligases can rejoin cut or broken DNA strands. In DNA replication, they join together the short segments of DNA produced at the replication fork into a complete copy of the DNA template. They are also used in DNA repair and genetic recombination. So DNA ligase is a type of protein. Topoisomerases are enzymes or proteins that have both nuclease and ligase activity. These proteins change the amount of supercoiling in DNA, so control its organization. Some of these enzymes work by cutting the DNA helix and allowing one section to rotate, thereby reducing its level of supercoiling. The enzyme then seals the DNA break. That's the ligase activity. These are required for many processes involving DNA, such as replication and transcription. So nuclease cuts and ligase binds, seals together the molecule back into its original, or into the continuous molecule. So helicases are proteins that are a type of molecular motor used. They use the chemical energy in nucleoside triphosphate, predominantly adenosine triphosphate, which is a type of nucleoside triphosphate, ATP, to break hydrogen bonds between bases and to unwind the DNA double helix into single strands. The helicases are enzymes which are essential for processes where DNA bases are needed. Polymerases are enzymes that synthesize polynucleotide chains from nucleoside triphosphate. Or such as ATP. The sequence of their products is created based on existing polynucleotide chains, which are called templates. It's function by repeatedly adding a nucleotide to the 3' end of hydroxyl group at the end of the growing polynucleotide chain. As a consequence, all polymerases work in a 5' to 3' direction. A nucleoside triphosphate is a molecule building block of both the DNA and the RNA containing a nitrogenous base found or bound to a 5-carbon sugar with three phosphate groups bound to the sugar. So these 3 phosphate groups are cleaved when you need to um, um, when you need to transcribe the molecule. They from DNA to RNA, 2 of the phosphate groups come off, and with the energy that they release, you have the mRNA chain, which is formed the lacking 2 phosphate groups. So instead of DNA, you get RNA. Instead of see diphosphoribonucleic acid, you get just ribonucleic acid. Many DNA polymerases have a proofreading activity. Here, the polymerase recognizes the occasional mistakes in the synthesis reaction to the lack of base pairing between the mismatched nucleotides. If a mismatch is detected, a 3 to 5 exonuclease activity is activated. The incorrect base is removed. In most organisms, DNA polymerases function in a large complex called the ribosome that contains multiple accessory subunits. So it's a molecule or protein that serves the purpose of cleaving and proofreading the molecules of DNA, and so the subunits include DNA clamp and helicases. Genetic recombination via chromosomal crossover is when 2 DNA helicases break or swap a section of the DNA and then rejoin it. This is how crossover occurs. This is important, especially in sexual reproduction, when you have to recombine the genetic material of each chromosomal strand from each parent in order to produce a unique DNA strand of the offspring. Structure of the Holliday junction intermediate in genetic recombination involving 4 separate DNA strands. Recombination allows chromosomes to exchange genetic information, producing new combinations of genes, increasing the efficiency of natural selection, and is important in evolution of new proteins, and therefore changing the organism's function. Genetic recombination can also be involved in DNA repair, particularly in the cell's response to double-strand breaks. So various functions of the cell can be affected by genetic recombination and the production of new proteins. Recombinases are enzymes which catalyze recombination reactions.